Maarten, je staat nog op mute.
Sorry about that. It's a first webinar after the holidays, so I was still muted. Thanks for pointing that out. So welcome to the NCOH monthly webinar. My name is Maarten Hoek. I'm your communication manager for NCOH and your host for today. This meeting will be recorded and made available through our YouTube channel. Today we have a special with 4 experts, and this webinar will therefore last 30 minutes longer. We also have a graphic designer, and the drawing will be shared during the panel discussion. And word has spread. Even though we had a holiday break, we have over 170 registrations from countries far and wide. And I'm just going to name a few countries to give you an impression how far and wide I mean. I mean Australia, all the way from Bondi Beach, that is far. Belgium, Canada, Cuba, Finland, Italy, Pakistan, Qatar, Somalia, South Africa, Turkey, UK, and all the way from the US. And this means that some of you are very early risers and others are night owls. So very warm welcome to you all. We appreciate all the effort you're making to join us. As usual, we will start with our guest giving a 15-minute talk, followed at the end by a 30-minute plenary discussion. And if you have any questions, please use the chat function. You can also request speakers or topics for future webinars in the chat function. So our first speaker of today is Dr. Fabian Leenders. He is famous for tracing the source of the world's largest outbreak of Ebola to a single tree, a remarkable work. And he created the Leenders Lab, which objective is to map the epidemiology of highly pathogenic microorganisms. And now his biggest task of all is setting up the first center for One Health in Germany. His talk today is on hotspots for disease emergence. Dr. Fabian Leenders, welcome, and the floor is all yours.
Well, thank you very much for this very kind introduction. I'll start sharing my screen, I guess. Here we go. I hope you can see that. Should be fine, huh?
Yep, that works fine.
All right, perfect. Thanks a lot. So it's a great pleasure and honor for me to give a short talk here at this webinar. The topic I was given is the hotspots for disease emergence, with some aspects of the field and ecology, so the real work where you get your hands dirty. So I'll give you a quick run through of what we are doing. So just as a general introduction, we all know that zoonotic diseases are nothing new. Since the Neolithic transition latest, the zoonotic diseases are hitting us, and some are also staying with us for long periods of time, as we are currently experiencing. For some of those diseases, we know quite well the origin and how the jump from animal to humans happened. For others, we are still in the dark. As you're probably all aware, the hotspots for these disease emergences are in the tropics, in those areas with high biodiversity. And this is no surprise, because this is where we have intense human and animal interfaces, where we have the according biodiversity, which we can see in the mammals and in nature. It's also reflected in the microorganisms, obviously. We have, unfortunately, significant climate change on the local and the global level, and this is really something in my 20 years I have been working in the tropics, had to experience very drastically myself. This is obviously linked to habitat destruction in according areas. What's new is the high mobility. So every little village in Congo Basin you reach these days with a motorcycle within a few days. This did not used to be the case in the years before, and leads obviously to a higher risk of diseases to spread from those remote areas also to the rest of the world. We obviously have the important component of population growth, which is significant in many of these areas. And unfortunately, this is coupled with very weak health infrastructure. And this means when a new virus or bacteria or parasite hits the human population, it's extremely unlikely in many regions that it will be detected, and it can spread undetected much further than, for example, SARS-CoV-2 was able to do before it was detected then quite early by the Chinese colleagues. So if we want to study disease emergence, zoonotic disease, we need a One Health approach. That's quite obvious. You have to look at the environment and human health and animal health all together. We have to lose the habit of using a tunnel view in research. What we are doing in my team mainly is to look at the relevant pathogens to try understand disease emergence and also to learn from the past. So we do the field work ourselves with our African partners, always in very close collaboration. So we go to the according areas. It's not always easy to get there. This is our long-term field site, or the road to our long-term field site in Ivory Coast, Côte d'Ivoire, where we're working since 20 years on humans and animals. We are also working in Congo Basin, where it's actually quite comfortable, but very expensive to charter a plane to get to where you want to work. And once you're there, you should not have forgotten any little syringe or anything, because you will not be able to buy it on site. And once you do the field work with all the data collection, sample collection, together with the local population, there is a whole bunch of lab work. And I'll not go into detail here, but they're all the classic methods these days very much shifted towards the next generation sequencing methods and also multiplex serology. And after that, you have the whole bioinformatics, phylogenetic statistics analysis. I will also not bore you with these parts here. So what do we do to identify relevant pathogens? I'm a big fan of surveillance. I think we need more real-world data, and we cannot only base our models on the few data we have. So we have two approaches. One is to really work with the local population, the highly exposed people, for example, looking at the bushmeat hunters here in Congo Basin, and then at a range of pathogens transmitted between the animals and humans. One example here was looking at the simian T-cell lymphotrophic virus transmission from the primates to the hunters, and then looking at other cofactors such as age, sex, activities, and so on. This is just one example. We do that for many different pathogens. Besides that approach, which you may have heard about before, which is quite classic, we also developed a little bit an innovative approach, which is to make use of our closest relatives living in those hotspots for disease emergence, sort of as natural guinea pigs running around. So they live in those habitats where there is probably a very high diversity of microorganisms, or there is, it's not probable. And among those, there are probably also some pathogens which may be of high relevance to us. So our closest relatives, they allow us to filter out the relevance. So by observing them, their family structures, knowing what they're eating, how they're exposed to the environment, and then following them individually, we can detect quite a number of relevant pathogens. So here you see, for example, the members of the South Community Thai Chimpanzee Project. We know them individually since 40 years, so we can observe their health without catching them, without interacting with them. But when they're sick, we can collect feces and urine samples. And when they die, we do necropsies with full PPE to find out what killed this animal. And whatever killed the animal is probably of high relevance to us as well. One example is that we found a new type of anthrax. It's a Bacillus cereus, which actually causes anthrax. So it's nothing you would ever expect to exist. And we did a number of series on these studies on these pathogens. And in the end, we could find that it's actually a major driver of wildlife mortality in the region. And by now, we also know that it matters for people in the region. It's not causing pandemics or epidemics, but it still kills those people who go in the forest and eat whatever they find in the forest. We found the pathogen first in Thai, but now we know it's widely distributed in a variety of species, not only the great apes where we found it first, also elephants, duikers, goats, and so on. So this is how the chimpanzees showed it a new pathogen of high relevance. We are also using innovative tools to monitor active outbreaks and diversity of the pathogen, for example, by using blowflies. Those flies find every dead mouse in the forest before you will ever find it, and they sample for us. So you just need to catch those flies, and that's a very useful tool to assess biodiversity, but also microorganism diversity. The second thing we do is to try to understand disease emergence, and this is done in our long-term cohorts, but also ad hoc if an outbreak is happening. And it's been mentioned already that I was leading the study or trying to find the source of the big Ebola outbreak in West Africa. And it's actually quite classic. What we did first was to try to find out if there was a large outbreak in wildlife in the region, because this is normally what triggers Ebola outbreaks in humans when you think about the history of Ebola outbreaks in Congo Basin. So we monitored those two forests in the southern part of Guinea, where we knew the animal density, so we were able to look at animal declines. We did not find any decline, luckily, so it must have been a different scenario, and the authorities informed us while doing these monitorings that they now knew where the index case likely lived. And this is Meliandou, this little village. So we went there to Meliandou, and together with the local population, we tried to find out what happened. And the best scenario, to make a long story short, was that the kids used to play in this hollow tree where there was a huge colony of free-tailed bats. And so you had a scenario like the caves in Uganda, where the bats are there, humans are there, and this is how they caught the Marburg virus, right? So unfortunately, we were not able to totally nail down the scenario, because the villagers burned the tree after the suspicion arose that they may have gotten the virus from those bats, which I sort of understand, being a family father myself, but it also destroyed my scientific career being able to pinpoint the real origin there. However, we have a good scenario, and these days people find other filoviruses in exactly this bat species. We find antibodies and so on. So there's probably more to the story than we actually thought ourselves initially. What we also do is to look in more the long-term cohorts for outbreaks, and then try to understand more cofactors, but I'll not go too much into detail here. And the last point I want to quickly mention is that we also want to learn from the past, because these epidemics, pandemics, they don't happen luckily every day, but they have happened a lot in the past. So what we do are two things. So I've shown that already, is to make use of the phylogenies of our closest relatives. And taxonomically, we are great apes as well, right? So we're looking at our cousins here. And for example, we look at different virus, human adenovirus C, co-diverged very nicely between the host and the virus. So you find that co-divergence. However, if you take the cytomegalovirus, a herpes virus, looking at the phylogeny, you will see that in the cluster of the chimps, there are also gorilla sequences, and in the cluster of the chimpanzees, there are gorilla sequences. So there have been interspecies transmissions, and we know from the field work with the primates that they do indeed interact in Congo Basin, in Gabon, and so on, and there are possibilities for transmission. So they show us that there is transmission possible. What we also then do is to look at archival sample. This is what my colleague Sebastian Calvinac-Spencer mainly does. He goes into the cellars of the museums and developed some methods where you can actually sequence full genomes from 100-year-old formalin-fixed samples. So that's quite cool. This is where he does his field work. And one little story we were able to publish beginning of, when was it? It was last year, was on the possible origin of measles virus. And he could, with the new sequences he derived from those old samples, nail it down further to the roughly 1,500 years back in time. And then working with people, historians, it's also interesting to see this is exactly when the human population reached the critical size needed to maintain measles virus in circulation, actually in the human population. So it doesn't help to alone look at the pathogen. You always take into account the other factors as well. Right, then I think I just want to summarize here that basically what we're looking at are very complex pictures. This is why I put up that picture of the Eidolon colony in Ivory Coast here, which is something extremely complex, hard to understand. And we need to look at all the different factors. We have to consider the past, look at environment, human health, animal health, and use modern tools to try to investigate disease transmission. Also want to briefly mention that we don't do that work alone. We heavily rely and work closely with our partners, in my case, from Africa, from politics to the master student. And this is extremely important for us. So we try to do most of our work in country, and that's a lot of fun and great pleasure. Also want to mention that it's the nature of One Health that you work with many, many different partners from virology and the top scientists on that side to the ecologists and conservation people. And obviously all that needs some kind of funding, which we get from various sources. If you're interested in what we are doing, you find more information on that homepage. And if you have any questions beyond what we can discuss later, feel free to contact me. That's it from my side. Thank you very much.
Thank you, Dr. Fabian. I mean, I think we could do a whole year with webinars with you and we'll still not be bored. It is quite incredible, all the things you have achieved already. I have a couple of scientific questions. I'm sure the audience as well. I have sensationalist question that I cannot wait. I've been meaning to ask you for a long time. How many days or weeks or hours even before you reached the tree was the tree burned down already?
Oh, it was just 3 days or 4
Oh.
days. Yeah, that was really disappointing. But yeah, I mean, the villagers really told us we had to gain their trust. They were part of the team. We asked them what they think happened. And in the end, they told us the story. They didn't tell us that right away. We needed to gain some trust. And I mean, I understand. I would have played in the tree as a kid. I totally understand. So I also understand they burned it.
3 days and 3 days. Yeah, well, it's still very impressive. For now, I think we're going to give the floor to the other guest speakers and we'll come back on the plenary session too. Thank you very much. So our next guest is Dr. Leah van der Hoek. She's a virologist at Amsterdam University Medical Center and developed a new method for virus identification and discovery. She was the first to discover the human coronavirus NL63 back in 2003. An endemic human coronavirus that remained unrecognized till then. It soon became clear that HCOV, or human, help me out here, human coronavirus, NL63 had spread worldwide with continuous reinfections throughout life. She also found several other previously unknown human and animal viruses. And in this webinar, she will talk about the technique currently in use in her group to detect novel viruses and the necessity to link a virus infection to disease. Yeah, welcome, and the floor is all yours.
Thank you, Martin. I will share my screen. Let me go back. Is it shared now?
Yeah, this works perfectly. Thank you.
Okay, thank you. Thanks for the introduction. I will tell, Martin already told, I will tell about the technique, finding new viruses, and I will combine it with an example of what we do when we find a new virus, because virus discovery can be that you simply characterize a lot of new viruses, but what we do in our team is also to try to get collaborations or to work together with experts or do whatever we can ourselves to link this virus to the disease where the virus is found in. So to start with is the technique. And then the example, the technique is VIDISCA. Virus discovery cDNA ALphaP. So cDNA ALphaP is the basics, and the main characteristics for this technique is that it's a restriction enzyme digestion to fragment the double-stranded DNA. And this is a technique developed by my group already in the early zeros. And that is the time when there was no next generation sequencing yet, which meant that we have to make this technique very, very, very sensitive to be able to find the virus, because we needed to clone everything in a plasmid and then sequence the clones after amplification. So we did a lot of optimization, which I will not go into detail because of time, but I want to show the overview of the method. It starts with the pre-treatment, and then every nucleic acids, is it RNA or DNA, is made into double-stranded DNA. And during this procedure of making everything double-stranded DNA, we use reverse transcription primers that will not anneal to ribosomal RNA. And by doing so, we really enrich this double-stranded DNA that we get when we make a library is largely enriched for viruses. And then that is why that's one part why the method is very, very sensitive. The second is the restriction enzyme and the ligation step. And I want to go into a bit more detail on that in the next slide. But to finish with, when we have done the ligation, and so we do a restriction enzyme digestion, we get sticky and nicely ligated. Then we can do PCR amplification, and then we sequence what we now currently do with the iron torrent S5. As mentioned, I want to go a bit deeper in one step because it's so unique. It's not used by any of the other library preparation methods that are bought or designed in-house. We digest our double-stranded DNA with a restriction enzyme MSC1, recognizing only 4 nucleotides, TTAA. And TTAA is AT-rich, no G's or C, which means that you have no problem when a target DNA is methylated. And recognizing only 4 nucleotides, which means that if the nucleotides would be randomly distributed, then every 250 base pairs roughly, there will be a digestion. And that is perfect for next generation sequencing, but also perfect to be able to see if this is a new virus or not. You have enough sequence information in 250 base pair to see if it's a virus we did not know of before. Also to see, by the way, if it's a virus that is known. So it can also be used for normal detection, but then very broad. This is then the next step. And the next step after this digestion with MSC1 is, of course, ligation to the anchor to be able to do the PCR amplification. But during this ligation, we keep the restriction enzyme active. I will see if I can have a pointer. Yeah, so here is the digestion, and then you here have 2 fragments, and they should not re-ligate during the ligation step. But of course, when you add ligase and you have these nice sticky ends, they will also ligate. So we keep the restriction enzyme active, and then when this is ligated, the restriction enzyme recognition site is restored, and it will be digested again. Whereas if this fragment would ligate to the anchor, then you end up with TTAT, which is no longer recognized by MSC1. So this is a dead end. And with the dead end, then you have a very efficient ligation step. So every fragment will at the end, you can leave it as long as you like, but at the end, everything is ligated to the anchors and you can do PCR amplification. And this makes the method very sensitive. And we measured that, this is already from a paper, I think from 2015 or 14. We measured it with all kinds of clinical samples containing known concentrations of virus. And then you see that from this on the x-axis, the virus load, that from 10,000 copies on per milliliter, which is only 1,000 copies in the sample that we test, because we take only 100 microliter, that we can start detecting the virus. And this is the percentage of virus read compared to the total. We tested it with all kinds of RNA, DNA viruses, and I already mentioned the sensitivity. You don't need millions of sequences. You only need 10 to 50,000 sequences. And we also do multiplexing, so you can do many samples on one S5 run and be able to detect the viruses. Then further to the examples, NL63 was already mentioned. That was the start of of my own group, I must say. So virus discovery was already, the method was started, then NL63 was found and was followed by several viruses. In brownish red are the human viruses, the viruses found in humans, and in black are the viruses that we found in animals. And it's not that these are the only viruses we found. These are the viruses of which we had the feeling that they are relevant. involved in disease, and only then we publish. And we always do more than just characterizing. We try to see if there is seroconversion, if there's an association with disease, all kinds of things to determine the association with disease. I will come back to that later by showing an example. Just want to mention here that Netwetwe virus is the virus that Arthur Edrich is going to talk about in the, I think, the next presentation. And the last discovery we did was circular single-stranded DNA viruses that we found in protozoa, which was published last year. And we were the first to find out that these protozoa were actually the host of these novel viruses. The example I'm going to talk about is the porcine pestivirus, which we published on in 2016. And that is a virus that we found in trembling piglets, piglets with congenital tremor type A2. And I have here a movie to show. To stop this, and then a movie to show the symptoms with some sound on it. You see that they are trembling. I don't know if you can see and hear, but they are trembling. Some have it more than the others. Some also cannot stand on their four legs. They have splay hind legs, and so they cannot stand on that. And this is in Dutch called trilbiggen congenital tremor. This is a movie from, I think it was from Korea. This is a symptom which is not found by us. This is a symptom which has always been known, already 100 years ago. There was a paper in veterinary medicine in 1922 where dancing pigs with these symptoms were described. This trembling is only in the newborns. These tremors diminish within weeks, but the main trouble is that they have difficulty to find the tets. Sometimes they cannot find the mother. You see that they can hardly walk when they have the splay hind legs. So they have the really high risk of starving to death. We had the hypothesis together with MSD Animal Health, where we worked together with, that this might be something infectious, because this giving birth to litters with congenital tremor occurs very often when the gilt, so the sow giving birth, is when it's a gilt, when it's the first pregnancy of the sow. And once it has done so, this doesn't occur anymore, as if the mother is at that time has protective immunity. So we thought it was infectious, and we even thought this way more when we saw outbreaks occurring in the Netherlands. And there was one farm with a lot of litters affected in the early 2012. So we had the hypothesis it's something infectious, it might be a virus. So I received serum samples from MSD Animal Health from an affected farm. And then I saw among the 27,000 sequences, I saw 30 sequences that had some identity to pestivirus, but it was not any of the known ones. And we did via full genome, via genome walking, we could determine the full genome. We also designed a universal real-time PCR to be able to screen farms with animals that were sick. And then we saw that there are many farms in the Netherlands infected. So here's the number of the farm. I don't know if you can see my arrow. But the number of the farms is there, and all kinds of types were found. And the virus was not found in farms that never had the disease. That's here assigned in controls. But we did see some positive piglets that had no symptoms, but they were from affected farms. Then we did phylogeny, and here you see that it's definitely a novel virus. I already mentioned the sequences we did. We saw some identity, but not completely identical to the known pestiviruses. And here you see that it's really a separate branch where APPV. By the time we were busy with it and did the animal experiments on the next slides, there was a publication describing this virus in in pigs without any symptoms as a common bystander, more or less. And that was the first paper on this atypical porcine pestivirus in 2015. So it's closely related to the other pestiviruses, classical swine fever virus, border disease virus, all kinds of viruses that are known to give disease in newborns, cattle, pigs, and sheep. The closest human virus relative is hepatitis C virus. So these are all the pestiviruses belong to the family Flaviviridae. As mentioned, finding a virus is one thing, but linking it to disease is step 2, which can be way more difficult than finding a virus. So we first determined if there's an association with disease, and indeed there was. We always found the virus in animals that had the congenital tremor, and we only found it in 9% of the healthy controls, of which some, as mentioned, were from the affected farms. So there were asymptomatic carriers in farms that had also the disease. Then the crucial question, can we reproduce the symptoms with experimental infection? And we did that in pregnant gilts, so the first pregnancy of a sow. Of course, we did it because the newborns, as soon as they are born, they are already affected, which means that infection occurs during pregnancy. So it has to be transplantal transmission of the virus. So we inoculated intramuscularly the pregnant gilts at 32 days after gestation. By the way, when I say we, this is mainly done at MSD Animal Health in Boxmeer. But forgive me, of course I use we because this was a collaboration. We saw replication of the virus in these pregnant gilts. Gilt 49 had a low concentration of virus in blood, and Gilt 50 and 51 had 10 to the 4th RNA copies in blood. So there it seemed that there was more replication of the virus. And then the litters were born. And what did we see? Well, we saw that Gilt 49 with the lowest concentration during pregnancy only had healthy piglets when they were born. And they were also all negative for the virus. But for Gilt 50 and Gilt 51, it was different. There were piglets with symptoms, 11 for Gilt 50 and 13 for Gilt 51. We saw that they were positive for the virus, so we could reproduce the disease. This was not done with cultured virus, but freshly obtained virus from infected piglets. In conclusion, we found the virus of which a new virus of which we not know if it's causing a disease, and we could reproduce the disease with the transplacental transmission. And this congenital tremor is most likely caused by this novel pestivirus. With the diagnostics we had, we see opportunities. Can we make farms free of the virus and thereby decrease this disease? We tried for years and published last year that we couldn't manage. So we tested a lot, and when litters were affected, they were removed from a farm. This is all in a closed herd. But after years, still the virus was there. So this is a very infectious virus and very difficult to get rid of when you have it on the farm. And with that, I end with that this is probably a very good target to be used in vaccination. This is my group at the Amsterdam UMC. I want to point out in particular the guy here in the middle. He's doing all the virus discovery when animals are the source of the clinical samples. And a very, very much appreciation for the collaboration I have with MSD Animal Health. This is a large list of names, but Arthur is really my companion where I work with doing the discoveries in the farm animals. Thank you.
Thank you, Dr. Leah. Very, very interesting and very nice to hear how much aspects are needed to understand before you have indeed an association confirmed. And that's one of my questions to you is, what is the timeline before you have a suspected infectious cause and you start researching until it's confirmed? And what are the main barriers in making that shorter?
The timeline is really when you have a disease and you see and you have a novel virus, then already the difficulty starts, because these animals never have one new virus. Every animal, especially animals, they have all kinds of new viruses. So you first need to make a selection, then you try to culture that virus, because pure culture is actually needed when you want to reproduce it. And culturing a virus is really a challenge. So that can take a year or 1.5 years if you manage already. And then setting up these animal experiments, 1 year has passed before you know it. And because we, my group and MSD Animal Health, are very, very picky, we don't want to simply publish on a new virus, but we want to say what it means when you're infected. So that's the reason why we can be scooped. So the virus is already published, but then we are the second publication saying what the virus is doing.
Yeah, so it's at least 2 years, but probably more, you're saying.
Yeah. I think it took us 4 years.
4 years.
Three, I think at least 3, but almost 4 years to have this completed, to have the paper published as well.
Yeah. Very interesting. Thank you. We'll continue with questions in the plenary discussion. Thanks for now. We're now switching to Dr. Arthur Edrich. I think we have misspelled your surname a couple of times in the social media we sent out. So let me pronounce it correctly. Edrich, right?
Almost.
Edrich.
Yes, thanks.
Edrich. Apologies, Arthur.
No,
Edrich.
that's right.
So Arthur, you are a medical doctor and you are almost or about to wrap up your PhD. And then after that, you return to work as a pediatrician at AMC, Amsterdam Medical Center. His goal is to become a pediatric infectious disease specialist and to bridge the gap between the advanced diagnostic assays available in high-income countries and high burden of unexplained disease in resource-poor settings. That is a good bridge. His PhD is on acute encephalopathies of unknown cause in children in Uganda. And Uganda is considered one of the hotspots for emerging infectious disease. And today, I believe you're going to tell us more about what you did in Uganda and the novel Orthopenia virus you found. Dr. Arthur, the floor is all yours.
Thank you so much, Marta, and all the other people that helped organize this. It's really an honor to be here and present indeed my work. I'm part, let me start my presentation in the meanwhile. I am part of Leah's group, as she already said, and indeed I've been working quite a while in Uganda, but also some other African countries to study the causes of encephalopathy, which is a difficult word for brain disease in children. And today I'll talk about a specific case. Actually, it's a story about the discovery of a novel Orthopenia virus in a Ugandan child. So a bit of a change of scenery again. We're going to move to Africa, where Fabian, of course, has done a lovely research as well. And now we're going to move to Uganda, which you can see in the green in the middle. And Uganda is quite a large country. So again, I'm going to focus in a little bit because we're going to a very small village with an extremely unpronounceable name called Ntwetwe. And just to get a feeling of what Ntwetwe looks like, and left you can see here the main road which is leading into Ntwetwe with a lot of trees, some small houses, people running around, and definitely no tarmac. And on the right, you see sort of more of the surroundings. So there's some forests, some swamps. So this is a very nice area for a lot of pathogens to emerge. But back to sort of where I want to start is actually it's about a 3-year-old healthy girl, and she presents to a health clinic in Ntwetwe Center, or in Ntwetwe, with headache, fever, and abdominal pain. And well, this is something that they see very often, you know, probably something malaria or something else. So they give her some paracetamol and antimalarials. However, over the course of about a week, she doesn't really improve. So we're back at the map again, and this child then, as the health clinic is not able to help her further, she moves from this infectious village to the nearest hospital, district hospital in Chiboga. And here she stays as well for about a week, but also here they're unable to determine what's going on and also unable to help her. And well, after deteriorating, they finally decide her to move her to the tertiary or university hospital center in Kampala, the capital center of city of Uganda. And that's also where I was at the time. So when she came to Kampala, this is sort of what we saw. So we saw a child again, 3 years old. She had a fever, an increased heart rate, an increased respiratory rate. So there definitely was something going on. And she was deeply comatose. She experienced generalized convulsions, seizures, decorticate posturing. Again, a quite difficult term, but that's something that you can see on the top right here. It's a symptom that's really indicative of severe brain damage where the cortical functions are being shut down. And then on fundoscopy, so when you look into the eye, we saw papilla edema. That's a sign that there's some increased pressure in the brain. So this kid, she was really ill. And then we started running the conventional diagnostics. So we made a list, what are the main causes of these kinds of encephalopathies. And one of the most common things is cerebral malaria. So she was tested for malaria, but she was completely negative. Well, bacterial CNS infections like meningitis, we tested for them with culture, PCRs, many other things, but also negative. Same goes for viral CNS infections. We ran a very broad range of viral PCRs, negative. And lastly, sickle cell, especially sickle cell stroke, is quite common in Uganda, but also she did not have this. And of course, there are other potential causes, but given the limited diagnostic capacity that's available in Uganda, this is sort of all that could be done for her in terms of diagnosis. And quite unfortunately, because we weren't able to diagnose really what's going on, we couldn't really treat her very properly, maybe, and there actually wasn't much improvement. And after a couple of days, her parents decided to go home again because there was no improvement, and it actually can be quite expensive to be in hospital. And very unfortunately, this girl passed away 2 weeks after. So that's not the end of the story. And as Fabian already beautifully illustrated much better than I can, Uganda, which is this sort of dot here, is sort of in the middle of one of the hotspots for both zoonotic but also vector-borne pathogens. And as Leah had already been discussing, we're very interested in viruses. So one of the hypotheses that we had is that, we initially excluded a viral cause, but maybe it's something uncommon or actually something new. So what we did is we used this method that Leah described, FIDISCA, on the cerebrospinal fluid, CSF, and the plasma of this patient. What we found was that there were 2 reads, which is quite a few, but still there were 2 that looked like a divergent Orthopenia virus. They were sort of 70% similar to viruses that we do know. So this, was already a sign, hey, this is a virus that it looks like something we know, but it's actually quite divergent from the stuff that we really know well. So maybe it's something new. And just to be sure that this really came from the patient, we ran a PCR for this piece of DNA or RNA that we found, and it was positive for both plasma and CSF. Now, later on, we also developed a quantitative PCR, and we were able to determine the viral load, which we could find both in plasma and CSF, and both was relatively low, but it was present. So probably this indicates that the patient had been ill for quite a while, and that the load may have gone down already by the time she actually came to Kampala. So I've been talking about this Orthopenia viruses, but what are they? So Orthopenia viruses are segmented negative sense RNA viruses. And with segmented, I mean that the genome is segmented in 3 parts. You got the S, you got the M, and you got the L segment. Orthopenia viruses are arboviruses. They're often transmitted by mosquitoes, and quite a lot of them can cause human disease. Many of them only give a very mild fever or asymptomatic even infection. Some are known to cause hemorrhagic fever, but also some are known to cause encephalitis. And this could very well be what actually happened with this index case. So we continued with genome walking. We were able to identify all 3 viral segments, although not the complete segments, but still quite a bit. And then we sort of could address the question whether this is something really novel. So to determine novelty, there you have the International Committee on Taxonomy of Viruses, and they give these criteria for when you can call something novel. And for these Orthopenia viruses, they say that the S segment should have an identity lower than 90%. As you can see clearly here, this is the sort of the closest ancestor. Tataguin has an identity of 59%, so we were quite confident that this is something novel. So here you can see the phylogeny of this virus. With here on the here in bold, you see nitrovirus again. And what you see is actually all these 3 viral segments, the S, M, and L, they all, for the phylogeny for each of these segments, the nitrovirus shares a closest common ancestor with this Tataguin virus. And what that means is that there has been no reassortment of segments, which sort of can happen with Orthopenia viruses, but that this virus by itself really is novel. More importantly, maybe, is that sort of the smaller clade of viruses that it clusters with, these are all transmitted by Anopheles mosquitoes. Hence also the name of the serogroups Anopheles A and B. So most likely this virus is transmitted by an Anopheles mosquito as well. And what we know also about other Orthopenia viruses, which I think is quite good news, is that humans usually are dead-end hosts. So usually there is some sort of animal reservoir, which could be a small rodent or a deer or whatever. And there's this cycle between rodents and mosquitoes. And it has like, well, two sort of life cycles. But then once it ends up in a human, it's a dead-end host and it cannot go to another person. So most likely human-to-human transmission is not happening. And then we want to go a little bit further. You know, we find this virus in the CSF, but you know, is this really, what does this mean? Is this causal? And like for the first step towards this, we sort of first looked at CSF. You know, are there any signs that this really was an encephalitis that was going on? And when we measured all these biomarkers, and these are the results, sort of what you find, what we found in the patient, and this is what you would find in viral encephalitis, and they all aligned. So indeed, it seemed that this was a true viral encephalitis, and as nitrovirus was the only virus we found in CSF, it was the most likely cause. But still, causality is always something which is very difficult to prove, and as Leah already showed, it takes a lot of time to do so. But I think even for human pathogens, it may be even more difficult. Of course, it's unethical to infect humans, especially when it's something deadly. You know, you don't want to do that. But also, you know, the very classic Hand-Koch postulates, they, for example, do not really allow that pathogens may also cause asymptomatic infection and only cause severe disease in a few. And also, for a lot of pathogens that we do know today, these postulates have not been fulfilled. So instead of trying to go for the Hand-Koch postulates, what I did is I sort of tried to establish causality criteria which are specific to Orthopenia viral CNS disease. So we looked at all the other Orthopenia viruses and all the causality criteria that are out there and made a tailored list. And I won't go into too much detail here, but for example, things that are important are disease association. For example, do you find a higher prevalence in cases than in controls? And where did you or how did you detect the virus itself? For example, you can imagine that if you do serology, that there may be cross-reactivity, but also that serology may not have a temporal association, as presence of IgG may indicate that there was an infection much longer ago. Well, so we also determined that other things like experimental infection, that the virus can be localized in the central nervous system, and others that they're important as well. So what we did is we looked at all the Orthopenia viruses that have at any time been associated with CNS disease, and evaluated sort of their, for all of these criteria, how well they performed. And we gave a total score ranging from 0 to 18. And we found out that there were a few which have really strong evidence of causality, which are the most well-known ones, like lacrosse virus. But for the one that were interested in tetravirus, it was pretty good, you know, but not perfect. It was moderate. So in some areas we did quite well, but there were 2 areas which I think we could improve. And I've shown them here, which are the association with disease, and secondly, experimental infection. So for the latter part, for experimental infection, what we tried at the Friedrich Loeffler Institute in Germany, I went there, we tried to inoculate rodents, but unfortunately this didn't work, and most probably because we only had a couple of microliters left of the plasma of the index case, which had been frozen and thawed many times, so the viability of the virus may have been impaired. But for the association, we had to go back to Uganda. So what we did is we did a case control study, and we thought, you know, where would this virus be? Well, most likely in the area where it was first found. Well, this patient at the time when she got sick, she was in the Twitch village, but this is a very small health center where it would be difficult to set up such a study, and Chiboga Hospital was relatively nearby. So that's where we did the study. So what we did, we had a case control study, including sick children, and each child will be paired to a healthy child. And at the same time, during the rainy and the dry season, we captured mosquitoes. And actually in all of these groups, we looked at whether this novel virus was in there. So this was a very big study. And well, these are 3 of the people that actually did all of our work here on the right. And over about a 1-year period, all patients with severe CNS disease, 2 patients with mild disease, and all of them matched to healthy control were included. Well, over 200 patients were included, incredible amount of work, but none were positive. So not a case, but also not a healthy control. No one was positive. And initially, this felt like a bit of a disappointment because, of course, you're trying to find this virus and everything's really, well, you don't know, and then you don't find it. But I actually, thinking about it again, I think this is actually quite good news because you know, this indicates that this virus actually isn't going around and causing a lot of children to be hospitalized. So maybe this should be positive. Well, that's actually what I think. Well, for the entomological part, the study team there, they captured over 50,000 flying insects. So even if Nitwitra isn't there, I think they captured all mosquitoes in Uganda. So all arboviruses are gone. So that's a good thing. But the samples have not yet been analyzed. So wrapping up, in conclusion, the tetravirus is a novel Orthopenia virus. It's probably an arbovirus as well, and humans are a dead-end host, so there's no human-to-human transmission. That's good. It was in the CSF of a Ugandan child with an unexplained encephalitis, and I do think this was really the most likely cause of disease. Well, there's moderate proof of causality, but the most important thing is it does not seem to be a common reason for hospitalization in that area. So there are many people I like to thank. There's a whole team in Uganda, also the patients, the caretakers. Of this, I really would like to thank Philly, who actually did all of the inclusions. Also Natalie, who spent a lot of time there. There's a team from Amsterdam, Germany, and of course the funders, but I don't have time to thank them as well. Thank you for listening. And if there are any questions, I'm happy to take them.
Thank you, Dr. Arthur. Super interesting indeed. And I'm not muted. No, I'm not muted. Good. Just checking. And I think you're right that it is good for humanity that you didn't find it. It was, of course, not very good for publications. So that's always a trade-off, I guess. Did you consider seasonality? Did you, those 200 kids that were involved, was it in a season or was it over the course of a year or what?
Yeah, exactly. No, that's a very important question. Yeah, we specifically decided to include over the period of a full year. Of course, Corona came around when we were doing this, so it was very difficult. But indeed, because of the seasonality and especially with the the rainy seasons, after which and during which you have all these mosquitoes coming up, you want to make sure that you cover those periods. So yes. You
did, okay. And did you try to find an animal reservoir as well for testing, or? So
we were initially planning. So the difficulty is, of course, where do you look? Where do you start? And the easiest thing in virus discovery is probably to look at the closest ancestors, because they probably share common features. That's also why we think it's a arbovirus. But for the animal reservoirs, it's a little bit more difficult. So we do think they're probably small rodents, but there are many rodents. And also that's an assumption. So we were planning to get some rodent trapping involved, but that actually was much more difficult than I anticipated. So we stuck with this, but that would definitely be something interesting.
Very good. Well, I'm sure you're going to get an award for eradicating all mosquitoes from Uganda. so excellent done. Okay, thanks for now. We're going to have more questions for you in the panel discussion. And now we're moving on to Dr. Chantal Bleker-Rovers. She has been working with the Radboud University Medical Center since 2007 and is an internist infectiologist. She's also professor of infectious disease outbreaks, and her research focuses on preparing for future outbreaks of infectious disease. That is a very hot topic at the moment. Specialized in Q fever since 2007 and founder of the Q Fever Expertise Center at Radboud UMC. And I believe today you will tell us more about Q fever. Dr. Chantal, the floor is all yours. Sorry, Professor Chantal, I should say. The floor is all yours.
Thank you very much. So can you see my slides?
Yeah, perfectly.
Okay, thank you. Yes, I will tell you something about Q fever, and we move back to the Netherlands again. And a little history. In 1935, Q fever was already detected in outbreaks among abattoir workers in Australia. And at the time, they didn't know what was causing it, so they called it query fever. 1 year later, Cox and Davis from the US found a new microorganism in ticks from the 9 Mile Creek in Montana. And Burnett also isolated a new microorganism from the patients of Edward Holbrook Derrick from the outbreaks of query fever. 2 years later, it was proven that those microorganisms were identical. And from the beginning, it was known that Q fever is a zoonotic disease resulting in outbreaks caused by the intracellular coxobacillus Coxiella burnetii. The bacteria are found worldwide. Most likely it is endemic everywhere, but in New Zealand, they have identified it from over 40 tick species, but also birds, deer, wild boars, hares, skunks, seals, sea lions, and even kangaroos. But also, and that's more important for human infection, in domestic ruminants, where they found a seroprevalence in cattle of 82% and in sheep and goats of around 73%. And it's also sometimes found in cats and dogs. Animals do not really get sick. If they're not pregnant, it's an asymptomatic infection. And the most important clinical presentation is abortion and stillbirth. And the abortion occurs at the end of the gestation period, and the animals do not show any previous symptoms. The bacteria are massively excreted during abortions, and during the Dutch Q fever outbreak, it was also found that comparable very high numbers of bacteria are also excreted during normal parturition in goats and also in sheep. The bacteria are also excreted in feces, vaginal mucus, and milk, but that's less important for human infection. And it was also shown that around 40% of pregnant goats on Q fever infected farms do have abortion, but the aborting animals recover rapidly and the milk production is hardly affected. So it's not really a severe disease for animals. Then I switch to human infection. Humans can get infected by mostly by inhalation of infected aerosols. And there airborne spread has been shown over a range of many kilometers, and already a single bacterium can cause seroconversion. No human-to-human transmission is very found very often, but during childbirth and with blood transfusion during the bacteremic phase, it can be transmitted between humans, and there have been some case reports about presumed sexual transmission. The highest exposure rates are found in people with frequent livestock context, such as 65 and 72% of persons living or working on dairy cattle farms are seropositive in Canada and the Netherlands. 74% of goat farmers are seropositive in the Netherlands, and 65% of veterinarians. There have also been multiple outbreaks described involving laboratory staff, but it can also occur in humans without any animal contact. And for example, in the Netherlands, many people live within 5 kilometers of a goat farm, often without knowing that they do. After human infection, 60% have asymptomatic infection and 40% have symptoms, and they should be diagnosed with acute Q fever. But symptoms can be very specific, like fever, headache, fatigue, sometimes also nausea, vomiting, diarrhea, rash, pneumonia, or hepatitis. In the Netherlands, around 20% of the patients were hospitalized, but in many outbreaks, that's below 10%. Mortality of acute Q fever is low, less than 2%. And you can diagnose acute Q fever by a positive PCR on serum with compatible symptoms. That's only possible in the first 7 to 14 days. Or you can use serology with at least 2 samples where you find a significant rise in titer or seroconversion. Patients can be treated with 2 weeks of doxycycline, and in case of an allergy or in children, you can also use quinolones, well, not in children, of course, or cotrimoxazole. After a symptomatic acute Q fever infection, 20% of the patients develop long-lasting fatigue and sometimes other symptoms, which is called Q fever fatigue syndrome. And in this syndrome, no active persisting infection has been shown. It can be diagnosed if the patient is still very severely fatigued more than 6 months following a symptomatic acute Q fever infection. There have to be severe limitations due to fatigue. Chronic Q fever needs to be excluded, and no other explanation for the fatigue should be found. We did a randomized controlled trial where we tried to treat patients with long-term doxycycline because there were some anecdotal evidence that there might be active bacteria still there, but it did not have any effect when compared to placebo. And we also tried cognitive behavioral therapy, which was more effective than placebo, but the effect unfortunately disappeared after 1 year. The patients with Q fever fatigue syndrome do not show significant improvement over time, and this disease has a very high impact on quality of life and work status. Of all patients infected with Coxiella burnetii, 1 to 5% develop chronic Q fever, and this can also occur after an asymptomatic infection. It can occur months to years following the acute infection. The longest interval we have found is over 9 years. And we're still counting. And over 50% do not have any symptoms until they present with severe and often life-threatening complication of the disease. The clinical manifestations of chronic Q fever are endocarditis. In the Netherlands, only 35% of all chronic Q fever cases and all in underlying valve disease. In France, for example, 2/3 of the patients present with endocarditis. In the Netherlands, we found more vascular infections, mycotic aneurysms, infected vascular prosthesis. Chronic Q fever in pregnancy is possible, but in the Netherlands, that was not a very large problem. It's probably strain related. And bone and joint infections are possible. Chronic Q fever is defined or diagnosed as a proven chronic Q fever when you have a high phase 1 IgG titer above 1024. you have a proven endocarditis or vascular infection, or a positive PCR on blood or tissue in the absence of an acute Q fever infection. Probable chronic Q fever are patients with a high anti-phase 1 IgG titer with risk factors for chronic Q fever, such as heart valve surgery, an aneurysm or vascular prosthesis, or symptoms of chronic infection or an atypical focus like bone infection without a positive PCR. And possible chronic Q fever are the patients who only have a high phase 1 IgG, which is not very specific. Patients with proven chronic Q fever do need treatment, and despite treatment, mortality is still high. Patients with probable chronic Q fever often need treatment, and some can be followed to see if they really develop a focus for the infection and then can be treated. And the patients with possible chronic Q fever only need follow-up. First choice treatment is doxycycline with hydroxychloroquine, and the hydroxychloroquine is added to increase the pH in the phagolysosome to increase the bactericidal effect of the antibiotics. This is a very long-term treatment, and it has many side effects, such as gastrointestinal side effects, but also phototoxicity that can be really severe and occurs in over 90% of the patients. And after a longer period of treatment, cutaneous hyperpigmentation can also occur. Alternative regimens can contain moxifloxacin or cotrimoxazole, or sometimes rifampicin, but we usually try to combine at least 2 effective antibiotics. For endocarditis, the treatment duration is at least 18 months, and in prosthetic heart valve disease, at least 24 months. The treatment duration in vascular infection is unknown, but in clinical practice, it's much longer. And even with this prolonged treatment, mortality is still above 25% in patients with proven chronic Q fever. Q fever outbreaks are very common. They occur around the world from a few to 400 laboratory confirmed human cases. It is estimated that the number of human infections is 10 times higher than the laboratory confirmed cases. Most outbreaks are geographically localized and restricted to one episode. And most outbreaks, especially the larger ones, are caused by sheep or goats. And there has been no real association between cattle farms and outbreaks, while the seroprevalence in cattle farms is high. And it's thought that cattle-related genotypes are less virulent for humans, but we really don't know why. In the Netherlands, we had an outbreak that started already in 2007 in a very small village in the south of the Netherlands, where a local family doctor reported an unusually high number of patients with atypical pneumonia. And after 2 or 3 weeks, it was known that all of these patients had Q fever. And as you can see here, Q fever is a notifiable disease in humans in the Netherlands. We had very low numbers of Q fever in the years before 2007, and then there was a quite steep increase in the number of cases. And as was shown later, we already saw that, I think I missed one. Yes, I missed this one, I'm sorry. The cause of the outbreak was goat farms, and there had been a strong increase in the number of dairy goat herds and goat numbers in the recent years in close proximity to highly populated areas, also located around the village where the outbreak started. So the epidemiological link was very clear from the beginning, but it took some time before this could be confirmed by genotyping data comparing microorganisms from the goats and human infection. And actually, this was the first large outbreak where the source could be confirmed by genotyping. And this table already shows that the 2 years before the outbreak started, there was an increase in farms with waves of abortion among goats and sheep. And when the outbreak occurred in humans, hygiene measures were taken in the dairy goat farms, and voluntary vaccination was performed, but this was not enough to stop human infections. And in the end, all pregnant goats had to be culled on Q fever positive farms, and mandatory vaccination eventually ended the outbreak. But as you can see, the outbreak lasted from 2007 to 2011. And it has become, unfortunately, the largest outbreak ever described worldwide, with more than 4,000 reported cases of acute Q fever, of which 9 people died. More than 44,000 people were infected based on seroprevalence studies, but other estimates are above 100,000. And more than 500 patients were diagnosed with chronic Q fever up until now. And in the last update in 2018, 86 of them died due to Q fever related complications. If you look at the costs of the outbreak, you can see in the table that the costs in the livestock sector are high, 85 million euros, but it's much lower than the costs in the public health sector. And most of the costs are due to income loss of people with Q fever fatigue syndrome or chronic Q fever. So to conclude, Coxiella burnetii can be excreted without clinical signs of Q fever in the herd. And acute Q fever can also occur in humans without any animal exposure because of the large the infection by aerosols that can be kilometers from the infected farm. Q fever outbreaks have a major impact on public health with severe long-term effects. And the costs are high in both the livestock sector and the health sector, but mostly in the health sector. Early recognition is very important to stop an outbreak on time. And you should look at abortion storms on farms and also unusual clusters of human atypical pneumonia cases. You need very quick source identification by epidemiological examination that should be confirmed by genotyping. You need a very fast response, and in that response you also need the One Health approach. And in the Netherlands this took some time before the cooperation was it wasn't ideal in the beginning. And in the Netherlands we have continued the vaccination of sheep and goats in highly populated areas, and this is very effective up until now in preventing new human cases. Thank you for your attention.
Thank you, Professor Dahl, very much for your presentation. I think we should go straight to the panel discussion. So if all the presenters want to join in by turning on their camera and unmute themselves, and then we can start to answer some questions. All right, I have a few. Thank you all for joining again. Thank you all for your presentations. There's a few questions coming in. Some are sent to everybody, some are sent just to us, so I'll try to address them all. I'm not sure if time, but we're going to try. The last one that came in is, why not vaccinate all sheep and goats?
Well, we do that now in the Netherlands. We vaccinate all sheep and goats on goat farms and where they have contact with the public, like in petting zoos and stuff like that. So that's still being done in the Netherlands, and that's something you should consider if you have large numbers of goats or sheep in highly populated areas. Absolutely.
Yeah, very good, very good. So I'm also very interested in if you have questions for each other. So please let me know if you have questions for each other. Fabian, do you think the blowfly would have told us what was happening in Holland or maybe in Uganda, in Kiboga, I believe, or Chiboga village before we could even detect it? I'm very interested.
In the approach to use the blowflies for monitoring. You know, I think there are interesting methods coming up to use environmental samples to also not only look for biodiversity. There's a nice paper out by another group which shows that if you just suck in the air through a filter in a zoo. And you do a meta-barcode and you can say which animals are living in the zoo by just looking at the sequences you find in the air. So I think we are underestimating the potential of such methods. And the blowflies are very obvious, right? They go on every pile of feces, you know that from the dogs and wherever. And they also find every carcass. And the frustration is when you work in complex habitat types, you don't see much, right? These rainforests, you cannot see far, but the flies will always find also a dead bat, or I think it's important to find out what kills those animals. So the blowflies help us a lot there. Not sure, you know, it's not a standard diagnostic tool, obviously, right? But it's kind of an indirect way of seeing, if you're lucky, the most prevalent things. So I think that's something to look further into.
Yeah, no, absolutely. I think it's very interesting. We also are doing sewage sampling, right, to monitor the COVID pandemic, which is similar-ish, I guess.
Yeah,
but it's super interesting. And I'm also wondering if any of you have any experience with citizen science, because, you know, now we are detecting new or potential new viruses or new infectious agents once there's many cases and there's a spike in reporting. But, you know, maybe we can move it forward like we do with the mosquito alert app. Perhaps you've downloaded it and they found a lot of tiger mosquitoes in Spain that way. Do you have any experience of working with citizen science and detection of new viruses?
No, not so far, but we are thinking in the same direction, trying to set up something. So with this new One Health Institute in Greifswald in the north of Germany, I'm thinking about involving like the students from school, the universities, to start a longitudinal, like systematic fly catching in different habitat types, and then look at antimicrobial resistance in that case. You can detect that nicely. And so that teaches them the thinking of One Health a little bit. At the same time, you're gathering long-term data if you make that part of their school program, which is quite a nice way, right? So we're thinking a bit in that direction, but the emerging viruses, really, that's, yeah, I think the mosquito people do great work there, especially when it comes to Europe, right?
Yeah, interesting. So between Arthur and yourself, I don't think we've got any flying insects left in a while. You sample them all. A question for Chantal. How do you suggest to practically implement the early recognition of clusters of pneumoniae? Technically, it's not notifiable, but in practice, clusters of pneumoniae or any other non-notifiable disease are rarely reported to public health centers. A GP might see just 1 or 2 patients with a cluster, and hospitals also don't see all the cases. Can we do surveillance from hospital and GP records combined? Question from Petra.
Yeah, that's a very good question. And I think we were lucky that this cluster occurred after the usual increase of pneumonia cases in humans during the winter season. And that there were so many, then it's quite easily recognized. But if you only have a few cases, then it's really difficult. So I think that's a very good idea to combine these cases. And I think it's also a problem because you will not find, for example, Q fever if you are not looking for it. It doesn't accidentally show up in your culture or whatever. So you have to think of it and you have to ask for specific diagnostics to find it. And I think maybe we should increase for our surveillance the number of samples that we test more extensively than we do for what's needed for clinical treatment. That's something that's an interesting idea. Absolutely.
Good. So I'm just going through some questions. The other question is, we've got lots of hotspots. You've identified them already. Most of you have spoken about these hotspots. Is there an estimate or a way to estimate what may still come out these hotspots in terms of future pandemics or other infectious agents? In which direction are you looking, and how would you assess that? Is there some kind of biodiversity and accessibility of humans and villages, or how do you do that? Maybe one of you can tell us more about that.
Well, shall I just make a start?
Make a start, yes.
It depends heavily on who you ask, because people who like doing sequencing, they make these estimations that there are still millions of new viruses to come, and which is probably not absolutely not true. Because many viruses are really host specific, and they are part of a natural situation, actually. And I think, Arthur, I think you know what came out of this huge sequencing effort from US, and how many human viruses or potential human viruses finally were found among the, I don't know how many millions of dollars that were used for sequencing. I think you know.
Yeah, I think even billions, or maybe that was the estimate. But I think, well, there was definitely one virus they found. But it was very difficult to prove causality there. But yes, it very limited.
So there are really large efforts, if we're talking about viruses and not simply all pathogens that can go to humans, but if we're talking about viruses, there are really huge efforts, and a lot is sequenced, and all these viruses are probably, that all these novel ones are probably not, well, only one, which is probably infecting humans, and there the disease is questionable.
If I may add something, I agree. You know, I had in very beginning a student. She was catching mosquitoes and then put them on cell culture and see what grows. You find lots of viruses, but what do they mean? So this is why I'm a big fan of surveillance, like clinical surveillance, no matter if it's human or animal, because then you see what, it really gets into the new host and it does something. So I think we need to get our hands dirty and do more surveillance, and perhaps, the all modeling and so on is also interesting and helps us to perhaps focus on certain regions, but I think we need more real world data. That's the one thing. However, like the Global Virome Initiative, I think that's what you're referring to. I was a bit skeptical for many years, but now if you think about the importance of understanding viral diversity to decide on like more as generic as possible vaccines, for example, which are cheaper now to generate than it used to be and more feasible, then suddenly it makes sense to know as much as possible about genetic diversity of viruses. For that sake, not for finding, oh, this will be the next pandemic. You will never be able to say that, right? It has a different purpose suddenly, but it suddenly makes sense, which I quite like.
Yeah, this last purpose there, it's useful too. But concerning the question, what to expect, I think that indeed surveillance is the way to go.
But in a way, that means we are always running behind the facts, no?
To predict a virus, the new pandemic, you can look at genetic features of the new virus or type of virus you found and so on. You can never really predict it. I think what we have to learn is, and this is where surveillance comes in as a training tool as well. We have to understand that health is something global, and as long as we have extremely weak health infrastructure in especially the hotspots for disease emergence along these gradients, those things will jump over. You will not detect them early enough to extinguish the fire before it spreads far. So I think we should put our effort in strengthening health infrastructure, training the doctors better in detecting something, new symptoms, something unusual, recognizing that something is unusual, and strengthening the diagnostics around the world, I think will help us to, as I said, jump on these little fires before they turn into something too big to control. I think that's where we should invest a lot.
Yeah. Thank you for that. And going back to surveillance, Arthur, another question for you that was posed. Did you find anything else in terms of the child, in terms of behavior or living circumstances or interactions or anything at all that may have caused this child to have this virus while nobody else has it?
Yeah, well, that's a very good, but also very difficult question, of course. And you'd like to do very proper epidemiological studies to find that out. But I also do think, so the answer is no. But considering it's an arbovirus, transmitted by the Anopheles mosquito, everybody gets bitten, especially there, by Anopheles mosquitoes. I think it's more of a chance process, and it may be that it was a young child, and then that given all circumstances that because of that, maybe many more people get the virus, but they don't actually get the disease. So I think it's just risk and risk and risk and risk, and the odds are just very small. So I don't think there's anything you can do. Well, unless bed nets and vector control and these things, but these are things we should do in a much broader sense, but not specifically for this virus. It's quite difficult.
If I may add there, what we would love is a serological assay, because then we can see how many have been infected but did not get the disease. But we didn't manage to get enough. I don't know, Arthur knows better, but to get enough sequence information to express a protein. So that's the problem. Yeah. We had such limited amount of this one serum sample of this one child. That we only have limited sequence information and could not express the protein.
Yeah, so one of the hopes that we have is to find it in mosquitoes still and be able to get the whole virus or culture or something from there, and then create a serological assay.
Yeah. Okay. And to come back to the hotspots, is it fair to say that now we're in a certain stage of human development, of colonization of this world, that we're still exploring or creating access to other hotspots, and at some point when we've explored and made accessible every inhabitable place in the earth, that we then go into a different stage of epidemiology for virus emergence? Or do you have any future thoughts on that, or thoughts on that about in the future?
I think it's not only about going there. If you consider even the remotest areas, well, which I know a little bit in Congo Basin, you find villages. You have connectivity now with the motorbikes, very cheap motorbikes provided by the Chinese. So it's more about the additional factors which play a role. For example, hunting, the whole bushmeat hunting. The closer you get to human settlements, the less you find the large mammals anymore. But people still want the bushmeat, they still go hunting. If it's for themselves and the family, that's something to really discuss very carefully, because you cannot take that away, and there are traditions and things like that, so careful there. But if you think about the big bushmeat market to the big cities and so on, people start to hunt more and more the small mammals now, so bats and rodents and so on, extremely species-rich taxa, which also means there is a different range of potential zoonotic viruses in there. So this is just one example for the shifts we are seeing. So it's not only about more people going to more places. There are many, many other factors in there. So that's why I think we will see more of those emerging diseases. I'm not a big fan of any predictions and prophet or whatever, but just the factor alone that when the virus hits, in old times, it hit a little village, Ebola, for example, then half of the people die in the village, which is really bad. Perhaps they make it to infect the next village, but then the virus could not travel. And the last Ebola outbreaks you saw in Congo Basin now and in West Africa anyway, the virus travels so quickly through the big cities now and so on. So this connectivity and enormous human population, coupled with the bad health infrastructure, as I said a few times, that's a real problem we see there.
Yeah. Could you tell us a bit more about the trade in bushmeat? Because I don't think many people realize how enormous this really is.
It's very hard to put good numbers on there. There's a few examples of studies done in certain national parks and so on, but we're talking about millions of tons of bushmeat extracted from those regions, right? And so it's really not just the one monkey here or there. We are really talking about a huge market with a huge amount of money behind it, right? So that's not so easy, and you have to look at it in a differentiated way, as I said. So the family father going hunting for the family, like he did, his grandfather did before. that's sort of, it's a risk. That's not a no risk, but it's sort of something to tackle really carefully. But then the huge market, it goes all the way to Europe, to China, and so on. Live animal market is becoming even more active again now, because other regions of the world realize they want a zoo now with some animals in there, so they have to come from somewhere. There are many things happening there, which are really important. And then it's about also, how do any people perceive animals? How do they handle them? The huge cultural differences of what you do. Like we did a study in Brazzaville on a bushmeat market where people sold the bats live, the fruit bats, they're live in the cage, they take them up and then they kill them, they hit them on the head, and then they put them in boiling water, and then they continue processing them. So why do they do that the one step there? Was there a bad experience, or what happened in the past? Why people do that? So I think those are really interesting factors. Yeah. Some cultures eat animals found dead. That's highly risky. And some would never do it, right? So interesting aspects in there too.
Absolutely, absolutely. A last final question for today. The role of vaccines. Can we not just develop vaccines for all our risky viruses that we know? And what is the cost and the implications, and what role do they play?
I think it's a beautiful topic for a future seminar. Yeah, I'm in favor of vaccine.
Yes. Yes, but just to say, they're super expensive to develop, right? And takes a long time just to have that question answered.
Well, sorry to interrupt there, but it got cheaper now with RNA-based vaccines. They're much cheaper than they used to be. And if you consider the costs we have now with the corona pandemic, we are looking at things from a different point of view, right? So yes. I'm curious how that discussion will develop. I totally agree to Leah that that's an interesting topic for another webinar, which I would then listen to and not be a specialist to comment.
All right. Well, that question came from Steph de Smet. So Steph, thank you for that question. We're going to have a next topic ready on that webinar. Rebecca made a graphic design based on the discussions and presentations that you all gave us. Rebecca, do you want to give us a short walkthrough?
Sure. Hi there, sorry.
Sorry?
Hello there. On the screen, you can see you should be able to share. You'll need to pin the discussion that you've just had with the question and answers. Can you see that?
Yes, I can see. So everybody has to click on the 3 dots above the green screen, and then you can pin it, and then it becomes enlarged on your home screen.
And all of the other presentations, the illustrations of those have been sent through, and so you can share them with your participants and your attendees.
Super, super. Really appreciate it. Thank you very much for this. So we'll be sending this around for everybody to study and enjoy. And I think with that, unless there's a final thing I'm forgetting, yes, I am forgetting something. Apologies. The thing I'm forgetting is the next webinar will be on the 7th of October. And it will be on the tripartite with WHO, FAO, and OIE on zero rabies by 2030. If you want to stay informed, please subscribe to our newsletter. You can also subscribe to our YouTube channel. And that's it for today. And I really would like to thank our speakers for today, Chantal, Arthur, Leah, and Fabian. It was really, really interesting. And Arthur, good luck with your new job. And with that, this NCOH webinar comes to an end. Thank you for dialing in and hope to see you next month.
Thank you, Martin.
Bye.